So Thea Norman is the strategy director at DREAM, and she plays an essential role in pretty much every challenge that we do, besides her other functions at Sage Bionetworks. She couldn't be here today, but she will have presented this. So we discussed this. We get data, crowdsource it, predict, but we know the ground truth. And we discuss data democratization, evaluation, and acceleration of research and collaboration. But how do we do all this? And we went through this process, and I want to discuss a few of the things that we have to take into account. So first, the scientific question. So this step usually involves coordination with the steering committee of experts. It's a mistake, I believe, to think that we, who are maybe experts in network inference, might know how to do a challenge on toxicogenetics. So when we did a challenge on toxicogenetics, we got together with the leads of the National Institute of Environmental Health Sciences, which in the US is the NIH Institute for Toxicology, and they proposed questions, and we guided them with our experience as what question is best for a challenge context. So, important thing then, we have to be aware of what is needed in the field and what are the questions that are the most pressing. We need to assemble a team, and this is easy to say, right? Thankfully, really thankfully, in DREAM, we have a number of, over the years, many people participated, and those people now are ready and interested in being part of the teams. Besides that, in our own institutions, Julio has technicians or students or postdocs that, and myself and everybody else, and at Sage there are people who are interested in participating in or the organization of challenges. We put together a team that usually has challenge director, which is people who are very familiar with challenges. For example, Julio is a challenge director, Pablo is a challenge director. We have a few others. Jim Costello is a challenge director, Lara Mangravit, and then Mike Kellen and Thea Norma. Anyway, so there are challenge directors that could, so to speak, direct the orchestra. But then there are challenge leaders, data scientists, and so on. So we have to put together all these challenges in a way that we have IT taken care of, data, scoring, statistics, and so on. Only then can we move on to the, making sure that the data we get is sound, that the data doesn't have too many missing values, and if they do, how to impute the missing values. We need to organize the data in a way that is easy to digest. If you want to do a challenge, you have to lower the entry level for people who want to participate. Therefore, you have to make it so easy to participate that at least some aspects, I mean, it's going to be difficult to solve the question, but at least reading the data and ingesting the data in your program shouldn't be a problem. There is an important thing here, which is the data governance. So you have to have agreements with data producers. We have to have institutional review boards. If this patient data, this is sensitive, you have to make sure this is de-identified and so on. So all this takes a while and needs to be taken care of. Then we do the dry run, which is a fun part of the challenge, in which we do the challenge ourselves. The data side, the director of the challenge works with the challenge leader to coordinate the data scientist to basically try to solve the problem and create a baseline that we hope, if we try to solve it in 1 month and we did this well, suppose, a little bit better than random, then people have to do at least that well. So if people don't do at least that well, the challenge doesn't have a winner. We will learn the level of difficulty in answering the questions, the deficiency in the challenge data, whether there is enough information, and the definition of a baseline solution, as I said before. We need to have a sound IT web infrastructure for which the Synapse team has, over the over the last few years, improve and improve, and I think that we have a great challenge platform within Synapse. Synapse is also a collaborative platform that can be used for other things. So I think that we have a great team put together there. They have web services that deal with registration, real-time challenge leaderboards, discussion forums, formation of teams, online supplement for challenge papers, wiki pages, and so on. Hi, we are so few that we introduced each other. What's your name? Eugenio. Welcome. Yes, so we have a website, dreamchallenges.org, where we document all the past challenges, community stories, who won the previous challenges and so on. And this is in coordination with Synapse. So usually you click here and you go to Synapse where the data is stored. Incentives. Let's discuss a little bit. What do you want to take a picture of?
Well, just for my wife. Do you mind?
No, that's all. No, just in case.
All the materials are also available.
I know it's like
oh, for fun.
Oh,
yeah.
She knows I'm working at the time with her. It's like, she's blaming that I was just drinking coffee, and I thought...
Go for it. No, I thought that you wanted to take some. Okay.
Sorry.
No, that's good. So, incentives.
Do it about it.
So, who knows what the incentives are? Well, I'm putting here, but let me go back. So, so if I give 4 incentives, what is the best incentives? The incentives are money. There are challenges with cash prices that have gone up to $50,000 in the past. But we are organizing a challenge for $1 million, but I'm not telling. I would like to participate in that one. But money, the opportunity to publish in a high-profile journal, the opportunity to have data that otherwise you wouldn't have pre-publication, the opportunity to collaborate and interact and have visibility through presentation in conferences. Which of these would be the most desired, do you think? The paper, right? Yeah. Because that's how we think in academia, right? Unless you get to $1 million probably. But so that's the question that we asked in several surveys. And so let's see how to break this. So the opportunity to publish a paper on your model in a top-tier journal is 1.6, 1 being the maximum. Opportunity to work with unpublished data, having access to data that hasn't been milked yet, is a second desired. Opportunity to speak at Dream Conference and get some recognition is the other one. Cash prices, however, is 3.8. Remember, this is 1 through 5, 1 being great. Opportunity to post a blog about your top scoring model doesn't look to be too desired. And I was talking before about challenge-assisted peer review. This is the concept that what better way to test a method than seeing how it fares with respect to other methods in a blind data set. Actually, we have met, right, Eugenio, in the past? No, I see your face. It looks familiar. No?
What might be missing is the networking to join future grants, projected grants. So whenever you join, you make investment for the future. And so on. You have ideas like how this could pay off. And the only thing they don't have is clear payoff is about publication. But it's the grant applications that you can start among your peers, not me.
Let's discuss that later. I think it's a great idea. We don't have it. We have something like that, but not that. It's a great idea. Okay. So these challenges is peer review clearly goes in the direction of publishing by participating in the challenge, and in that sense, people like that incentive. These are the journals that we have published with: Science Translational Medicine, Nature Biotechnology, Nature Genetics, Nature Methods. Actually, we already published Nature Methods, Nature Neuroscience, PlusCom Bio, And the National Cancer Institute, and this is one thing I wanted to say, Stefan, is that National Cancer Institute has given and is going to give in the prostate cancer and gave in the NCI Dream Cancer money to the best performers. So we say, you are the best performer, you must be good. It's challenge a CCTR review. I don't need a reviewer. The review was the challenge. Here, $50,000. And in the prostate cancer challenge, $75,000. For
It's a cash prize, it's a grant.
it's a grant.
Yeah.
It's a contract. It's a
But this is the kind of one kind of not the one on the here,
but it's a different one. But it's okay. This was incentives, advertising and communication. You know, this is something that we always try to think how to get the word out. Press releases, mailing lists. We have a lot of research. Probably many of you are in the mailing list, in the Dream mailing list, and we bombard you with some email. Sorry about that. But we need to reach out. Post advertising, banners. We try to advertise in Google Ads, didn't work very well. Webinars, word of mouth. We have the challenge open phase. We have a typically training set, not always, but most often. And then, you know, the leaderboard through the Synapse web services. I think leaderboards are great motivators to see yourself, how you're doing with respect to others, see whether you are improving as you are improving your algorithm. So I was going to talk about cheating, but let's not. There is a lot of cheating in these challenges because people are starting to get something out of participating in Dream challenges. For example, they get jobs. You know, it's a good thing to say in your CV. And it has motivated some people to go beyond what is strictly scientific and try to bend the rules. I was going to discuss this, but you can imagine different ways to cheat that we learned. For example, there was one person that cheated in one challenge, and I asked him what we discovered him. He didn't come to us. We discovered there had to be a finite number of submissions, and this guy had created like 10 different avatars, personalities in the web with different web addresses, URLs, and so on. I asked, why the hell did you do that? You almost screw up the whole challenge for all of us that organized and for all people who participated. And he said, yes, I did because I thought it would increase my chance of getting into a good PhD program. I wouldn't have done it if it was not for that in that situation. I just thought that application for PhD programs is really competitive, and I really lost my mind. I was unable to stay sensible. I became fooled by my stupid ambition. He's not an English-speaking person. But you see, this is the things that we have to make sure that doesn't happen because everybody's work gets jeopardized by these kind of things. Scoring, I will not talk about this because we will talk about this later. Then the analysis post-challenge, why some methods work better than others, where is the signal in the data. Sometimes we don't find signal. We recently finalized submitting a paper in which we show that we wanted to find, it was a GWAS kind of challenge. We had to predict whether there was, whether we can predict who responds to anti-TNF treatment in rheumatoid arthritis. Patients, we had from the patients baseline clinical and demographic information and genetics, and we thought genetics was going to be useful. Compare the submissions with genetics and non-genetics, no difference. Actually, sometimes genetics makes it worse because it has noise that confounds the clinical information. So the paper is really about, there is no data in genetics. So sometimes the signal is not there. And the wisdom of crowd analysis that usually we do.
So this type of finding, when it's spotted during the dry run?
Yeah, it was spotted in the dry run, actually. We didn't find a lot. We found a little, but not too much. And we said, well, we found a little, as we found a little, not statistically significant though, in the challenge. But because we didn't find the dry run, this is the dichotomy. should we run it so that people maybe find it that we didn't? But that's a good question. I was going to talk about the wisdom of crowds a little bit more. The fact that this is the wrong way to do it, as we said, because you know here, but there are other ways to do it in which you do a kind of cross-validation that doesn't use the actual knowledge. And also we see there that there is an increase in signal. I will discuss this in this afternoon's Wisdom of Crowds paper. Then we write papers. This is a little outdated, but we have 25 journal articles. Actually, if you count people that submitted their own methods, there are more than 100 people that use the data in DREAM or in their thesis or in their papers. In Google Scholar, we have over 1,500 citations to DREAM papers, PhD thesis, challenges in the classroom. We are trying to do it, and this is the first time that we are presenting this. Your feedback as to this was useful or not will be important. And publications.
All right, great. I'm just going to go over this relatively quickly. So how many of you in the room have signed up for Synapse? All right. How many of you have become certified users? Couple. There are instructions in the email that got sent out, and we can discuss this in a little bit as well. Has anybody used Synapse previously before this workshop at all? Okay, great. That's a good baseline to know. So just to give a little bit of background about Sage Bionetworks and sort of set the stage for why Synapse was developed. So Sage is focused on a world where biomedical research will fundamentally change to be more open and collaborative. We believe so strongly in this that we assume that this is true, and everything that we build, every collaboration that we enter into is with this in mind. We're a nonprofit, and we pilot a variety of components which are necessary to build a scientific research commons. Our president and CEO, Stephen, oftentimes talks about these pilots, and I think it's really important to understand that we run very high-risk pilots. We tend to do things that are a little bit outside of the box, and we do it with the understanding that we may not always succeed. It may not always be the correct answer, but we sort of see ourselves as we see running these pilots as a very important thing for the broader community, such that the community can learn what does and does not work. And at the end of the day, you can build infrastructure, you can build systems, but ultimately, the reason we're building these is to advance science. And so we support a number of communities of researchers who tend to work collaboratively across disciplines, and Synapse is one of those systems that we have built. So Synapse supports open scientific collaborations for clear, reproducible science. That's a lot of buzzwords, but I'll explain exactly what I mean by that. It enables sharing of resources, ultimately files, and those files can be data, code, and results, and importantly, their relationships to one another. And this is what we call provenance. So essentially, connecting the dots between the data, the code, and the results that are carried out. And it provides a mechanism to communicate in real time as opposed to only at publication. So just to dig in a little bit on the sharing of these resources, on the right here, this isn't a great graphic, but this is an example of a provenance graph that was carried out. You can see essentially the workflows that are carried out, who did the work on what date, and what the resulting object was, and you can sort of string these together to see what individuals have done. People can get credit for the work that they're doing, and you can start to build off of other people's work much more quickly because you can understand what they've actually done. It's important to note that there are a lot of web services out there that provide bits of this. We're not trying to reinvent those. So for instance, all of the code that I use when I'm running analyses, I store in GitHub. GitHub is an awesome tool for code versioning. We're not trying to reinvent that, but what I can do within Synapse, and I'll show this a little bit later this afternoon as we get into some of the live demos, is I can reference that. So Synapse is agnostic to where each individual file, each resource is stored. It's essentially a pointer to a URL, and we provide metadata around those. So you can use AWS, you can use Google Compute, you can use Figshare, any of these tools that are already available for different parts of the research process. And Synapse is ultimately a set of open RESTful APIs. Do people know what APIs are? Nods or shakes? No? So they're programmatic interfaces. So essentially what this means is there is an underlying architecture that can be called from a number of different, what I'll call clients. One of those clients is synapse.org, the website that you guys have all seen at this point. But importantly, you can access all of the same information that is displayed to you through synapse.org via programmatic clients. And so that means that all of the data, all the metadata that is surrounding these objects within the system can be pulled directly into a programmatic environment. So we have fully supported clients in R, Python, and Linux command line. So essentially the most common analytical open source tools. And to talk about sort of the different components of Synapse, it's ultimately sort of a distributed inversion file system where you can richly annotate, provide metadata on all of the objects within the system. There are customizable schemas for structured data. So this is both within the scope of providing metadata around a file. So you can tag a file as being, you know, breast cancer data set or any sort of key value that you want to provide. But we also have the concept of structure within a data set. So we have a feature called tables where you can provide a schema of tabular data. So you can say this column is a string of max length 50, this next column is an integer, this next column is actually a file, and it provides a pointer to a file. And it becomes a very powerful way to be able to query within data sets and only pull down information that you ultimately need. And again, we're agnostic to storage solutions, whether that be the cloud, various cloud providers, a local server, etc. GitHub is another example. By default, so Synapse is built on top of Amazon Web Services. And by default, anything you quote-unquote store within Synapse is stored in S3. And then we provide sort of the access restrictions and the governance around that to determine who can and cannot see it. And I'll talk about that in a little bit. We also have rich wiki authoring, so you can provide narratives of the work that you're doing. You can embed videos, provenance graphs, images, LaTeX right into a web page. It's a very powerful way of communicating within a group of collaborators. And then we have access controls such that you can share whatever you want with whomever you want. By default, when you create a new Synapse project, you're the only person who can see it. Then you can invite individuals and or teams of people to do different roles within there. So I could grant Gustavo the ability to edit my wiki pages, but I don't trust him enough to give him delete permissions because he might, get rid of some important data that I have there. So, and on top of the access restrictions, Gustavo actually touched on this a little bit. We also have a governance system, which is very important when you're starting to deal with sensitive human data. When we're dealing with in silico data sets or data sets on cell lines, it's a little bit less important. But we essentially have another tier of restrictions, which we call governance restrictions, that can be placed on top of data that may require IRB approval to be able to access it, or other terms of use that people put on top of those data.
Well,
Gustavo?
I'm not sure whether in Europe they are called IRBs, boards that regulate how human data is going to be used.
Yeah.
They are called IRBs as well.
No, ethics committee. What was it? Ethics committee?
Ethics.
Yeah, so in the U.S. we tend to have both, actually. So IRBs tend to deal a little bit more with legality issues, and there are also ethics boards that deal a little bit more with protecting of subjects.
It's more about making sure that what you do is HIPAA compliant, and that what you're going to be doing with your data is sound, is not going to hurt patients. So these protocols, you have to make a proposal on the review board. So there must be some formal way of dealing with that. But I don't know the name of it. Hey,
Welcome.
another dream.
Another lecturer.
Hey,
So I think at that point, that'll sort of wrap up the high-level overview. And then when we talk about participating this afternoon, I think I can give a little bit of a hands-on, walkthrough of Synapse as well. So I think we're just about at quarter after, which is
We can leave a
coffee
couple of
break.
minutes for questions.
Yeah, questions?
could you extend, like, maybe we'll see it in a second, but what can happen when we have lots of data on our patients and that's right. And if I would like to tap into the workflows we have come up with, should I upload those, or should I just download the workflows, or what is the kind of overall scheme that you see for some clinical intelligence science?
Sure. So I'll say that Synapse is not a workflow engine, and this is something that we can talk about a little bit later. It differentiates itself from something like Galaxy, which is a little bit more workflow motivated, whereas Synapse is more focused on communicating what you've done. So those provenance graphs are really just an analyst's description of what they've done, as opposed to a hardened workflow with inputs and outputs, if that makes sense. You can get to the underlying code, but there's no guarantee that there's no Synapse stamp of approval that if you take this data and you run this algorithm, you're going to get this output. It's really more of a communication tool than it is a hardened workflow system. And so Synapse does not provide any compute. That's something that you do locally, and then you sort of register with Synapse what you've been doing.
So the expression data set,
Mhm.
we upload to ArrayExpress or Geo,
Mhm.
whatever, and I give a reference to that, or is that I upload to you directly?
You can do either. And so I can provide some examples, in fact, this afternoon if you want, where I have linked to a Geo data set, for instance, or an ArrayExpress data set that is stored there. It's not stored within Synapse. But once you register with Synapse, one of the nice things is you can programmatically access it then, and you can use it in those provenance graphs. And so you can use the Synapse analytical clients to go and fetch it, even though it's not stored within Synapse. The actual data is, and just the metadata is. So it would go to ArrayExpress, or it would go to Geo. You pull it down locally, do whatever analysis you want, and register the result with Synapse.
So you read on all the invitation work of the source for the description of patient controls and description of phenotypes. You've done the same thing, or is it just a text file that I find?
You can do it either way, ultimately. You could either point to it, again, living elsewhere, or you can actually upload it yourself.
Can you talk a little bit about, because I think it's an important function that Synapse has. I don't think you mentioned the Bridge project, because I think it's very important in the direction of collecting big data.
Yeah, so one of the other software actually our other big software offering is something called Bridge Server. Has anybody in here heard about the Apple Research Kit APIs that were launched earlier this year? So it's a set. So Apple, when they announced the watch, when they had their big event in March this year, leading up to that, they announced an open source project, which for Apple is a little bit different. They're not known for being very open, but they announced an open source project called ResearchKit, which is a way to conduct research via mobile phones. And in particular, the first iteration is through iPhones, but they actually made it an extensible platform such that Android developers could plug into the same framework. And so there were 5 studies that were announced at that time. Sage Bionetworks is leading 2 of those 5. The other ones are being led at Mount Sinai, where Gustavo has an appointment, at MGH, Mass General Hospital in Boston, associated with Harvard, and at Stanford University. All 5 of those studies are recruiting participants and pulling data off of mobile phones through ResearchKit, and that's being transmitted through something called Bridge Server. All 5 of those studies are using Bridge Server as a backend. And then Bridge Server is separating out account information and participant information. They're providing a unique health code for each of the individuals, such that there's a coding process, such that there's less identifiable information provided to the researchers. And then the data is ultimately made available through Synapse for the researchers to consume. And the types of data that are being collected, there are sort of 3 classes of data. One is surveys, so they're very classical question-answer, survey methodology, which has been around for decades and decades. The second is activities, active participation, and so an example of this is in our Parkinson's disease app. We ask people who have Parkinson's disease before they have medication, directly after they have medication, and then long after they have medication, to do a tapping test. And so they see within a, I think, 30-second or 60-second window how often they can tap back and forth alternatively on 2 buttons on their phone. And so it's something that they're actively participating in, and we collect all sorts of metadata around the rate that they're tapping at, the placement on the phone where they're tapping at, so are they tapping in the same place, are they all over the map? And then the third class of data, so we have surveys, we have active participation. The third is passive information, and this is things like how much you're moving during the day, the step counts, the accelerometer data, relative GPS. We don't collect absolute GPS, so we're not tracking exactly where everybody goes, but we can tell relative to a fixed point where you're starting, how much are you moving around during the day. And there are other passive tasks that can be collected on the phone as well. And so we're doing this across a number of different diseases and afflictions, and it's, as Gustavo mentioned, a lot of data. And I think it's our hope in the future that some of this data will be made available for a dream challenge as well.
Let's see, this is very new and not used at Synapse, in which, how do you store this data that people are collecting? And Synapse is a perfect platform for that, because on one hand, it identifies and enables people to directly deposit their data there, and then on the other hand, it makes a bridge. That's what it's called, bridge, for that data to be used by researchers. That otherwise, don't know where to go. But in this case, all the data will be collected there. So I think it's a very interesting and new way of working.
I think one other thing that I'll just mention quickly, which I think is very intriguing, is that for all 5 of these studies, participants were asked at consent time if they would like their data shared with the study research team, so just the team who's carrying out the study, or if they would like their data to be made available more broadly to any qualified researcher. And I think it was around 75 to 80% of participants said they would like their data to be shared more broadly. And so what we're hoping is that within Synapse we can start to build this bolus of data, this commons, where people can start to come in, not just the people who are conducting the research study, but other people who are interested in these types of data, to be able to build algorithms and provide insights out of it. So